Takanori Ogawa joined Certara in 2010 through the acquisition of Bellkey Science Inc, which was a distributor company of Phoenix products in Japan. And now, as that is AKT Health Analytics, is the exclusive distributor of Certara's products here in the Asia Pacific region. During his time at the companies, he has provided scientific support, training, and services for PKPD modeling and simulation analysis to scientists in the pharmaceutical industry, academia, and regulatory agencies of Asian countries such as Japan, Korea, and China. He has also conducted over 300 training courses in those countries. In the courses, he teaches scientific theory and practical applications in addition to software. Hands-on exercises so that attendees can really practice the M&S approach to their own R&D project. Takanori earned an MS in Environmental Science from the University of Tsukuba. Taka-san, welcome to the GVHS 2022. Thank you so much for taking the time. I will now give you the access to share the screen.
Okay, sure. Thanks, Rashmi. Oh.
Yes, you should be able to share your screen.
Okay. So can you see my screen?
Yes, we can.
Okay. Yeah. So thank you for having me in the virtual summit. It's an honor and a privilege for me to present our software solution to everyone in this event. So my name is Takanori Ogawa from Certara, Japan. I've been working at Certara for 12 years and providing scientific and technical support to our customer in pharma, especially in APAC region, including Japan. And today, I'd like to introduce our RsNLME software, which is a software solution for population pharmacokinetic and pharmacodynamic modeling. And this mathematical modeling approach is an essential data science tool to find an optimal dosing strategy in the modern drug development project. So I won't cover the detail of the methodology today. Rather, we'll focus on how capable RsNLME is in your pharmaceutical research and its use case. And I'll try to address your questions at the last Q&A session. If you have any questions, please feel free to ask. Okay, so let me start. So first off, what's RsNLME? So how could it help you conduct your data analysis and build your career as a data scientist or pharmacometrician? So before diving into the detail of the software, let me present the latest achievement in Certara software business. And Certara provides a state-of-the-art software leveraging modeling and simulation technology to accelerate a drug development project for patients. And we released several software in recent years, and I believe they could be a big help for your success in your own drug research and development project. And the first two software, Certara Integral and Phoenix Hosted, they are our cloud-based solution, which provides a secured and validated environment hosted by Certara for efficient PKPD data management and analysis. And PK Summit automates a complex and time-consuming PK analysis workflow for regulatory submission to, for example, US FDA. And PK Summit ensures compliance with the industrial standard for CDISC data submission through Pinnacle 21 technology. But I won't cover more detail of these software today. If you want to hear more details, please reach out to us and our partner, AKT Health team in India. So RsNLME is a software for population PKPD modeling running from R. But what's R? R is an open source programming language and environment for statistical computing and graphics. It's a very popular language in the whole data science field, and a huge number of companies, including pharma, look for a highly skilled person in this language. And it's actively used in pharmacometric community already for data manipulation and visualization in M&S analysis, and recently SAS replacement or Shiny app development. So if you are not so familiar with this language yet, I'd recommend to start learning the language as soon as possible, because acquiring the coding skill could boost your career development and, of course, your current and future study. And with this respect, RsNLME could be a very powerful tool for you. So what is the capability of this software? It has a collection of all packages and companion RShiny app for population modeling. With these R packages, you can build and edit population model and run the model with multiple options like parameter estimation, covariate search, model validation, or simulation, and so forth. Also, the last three packages help you to generate a high-quality model diagnostic plot and data analysis report from your modeling. So by the way, why could our RsNLME be so powerful to learn our language? So you might be reluctant to use our RsNLME regardless of its capability, because it should require our programming for anything you need to do. Actually, our RsNLME uses tidy burst syntax, and you can write a code in a very efficient way. But if you had no coding experience before, writing these codes might be still very complex and challenging for you to see what to do for your study. Okay. For the case, our RsNLME provides a clear path to learn our coding for people who need assistance on it. But how? As I presented, our RsNLME has companion Shiny apps for every step in population modeling, like model building, execution, and evaluation. And these Shiny apps provide a graphical user interface or GUI over our console, and you can use our RsNLME functions in a very user-friendly way without coding. But it's going to be a very conflicted situation, right? You can do a modeling from all, but it appears you don't have a chance to learn how to code all. But no worries, actually, these Shiny apps can generate R codes from your mouse and keyboard operations on the Shiny apps like this one, okay? To ensure reproducibility of your analysis. And by reviewing these R codes, you can definitely get a chance to learn how you can write an R code to reproduce the same result on R. So what I want to emphasize here is our RsNLME is capable of supporting both high-quality data analysis and your learning of R at the same time. On the other hand, you might be concerned about the computation performance of our RsNLME. That is, could we trust a computation result from our RsNLME and use it for our regulatory submission or some key data analysis? And the simplest answers from my side is absolutely yes, because its modeling language or PML and NLME engine for modeling computation, both implemented in our RsNLME, come from Phoenix NLME, which is another population modeling software developed by Certara. So I'm saying that Certara provides two different modeling software using the same computation assets. And Phoenix NLME has been widely used in regulatory submission to, for example, US FDA or Japan PMDA in the last decade. In other words, the computation performance of our RsNLME has been thoroughly tested through Phoenix NLME, and that's why you can trust your result already. However, there is still an issue with our RsNLME operation, which is a management issue of your input and output data. So as you may know, in Phoenix WinOnLin, another PKPD software developed by Certara, in the software, every data is stored in a single project file. So with Phoenix Winonlink, your data is automatically managed in a very efficient way, but our RsNLME generates multiple folders and files for every modeling. So you would have a huge amount of data files through your analysis, which is very time consuming to manage. So it would be better for us to have a kind of workbench to use RsNLME more effectively. Okay, and Piranha can work as the workbench or electronic lab notebook for RsNLME modeling. And Piranha is another software developed by Certara. And as you may know, this software has been developed for non-mem analysis, which is a golden standard software for population modeling. And now it supports RsNLME analysis as well. And Piranha provides the GUI to facilitate the management of modeling results and has a great capability and flexibility to harmonize workflows in an organization and address individual needs of different modeling problems. Also, our RsNLME Shiny app and the library of our scripts are seamlessly integrated for further modeling works on Piranha. So, I mean, you can use our RsNLME alone, but I highly recommend to use our RsNLME with Piranha, which could maximize the productivity of your population modeling. So with Piranha, you can address the data management issue. However, again, you'd have less chance to learn R coding because you can complete most of modeling works from Piranha GUI, but you can still learn something from our RsNLME Shiny app and our script library implemented in Piranha. And when needed, you can write your own our scripts from Piranha to address your own problem. Yeah. So with that, I gave you a brief overview of our RsNLME and Piranha products in the previous session. So now I'd like to present a case study using Certara NLME solution, which I'm involved with recently. So in this case study, the ultimate goal is to develop our Shiny app for physicians to predict the transition of neutropenia in cancer patients based on Bayesian simulation using a population model, and decide the necessity of prophylactic GCSF administration to avoid severe incidences of cancer patients. Since the administration cost of GCSF is very high, so this study is expected to improve the cost effectiveness of GCSF treatment in hospital. And the study mainly consists of 3 steps. So first, we built a population model to describe the time course of drug concentrations and neutrophil counts. So you can build a model in our RsNLME, of course, but we selected Phoenix NLME instead, because when I worked on this modeling project, I was more familiar with Phoenix platform, and our RsNLME was not released actually. So next, we developed our Shiny app for Bayesian simulation, and our RsNLME was used to implement the best model selected in Phoenix NLME to inside the Shiny app. Lastly, we conducted a prospective clinical trial to evaluate the clinical utility of the Shiny app. And this trial is actually ongoing right now, and we are expecting to report the result in the next 1 or 2 years. And this slide summarizes more detailed background of this study. So first, we focused on amubicin for small cell lung cancer treatment, and PK exposure of its active metabolite, amubicinol, shows a correlation with the severity of neutropenia. That's why building a population model is meaningful to predict the transition of neutropenia for a specific dosing regimen. However, you might be wondering why we need to develop the Shiny app. So I mean, we can conduct a Bayesian simulation on Phoenix directly, But actually, it's not so easy for the physician who are not a software expert to execute the simulation on Phoenix. Therefore, we need to develop a more user-friendly interface for the simulation. And from this aspect, our Shiny could provide a great flexibility and assist the physicians to leverage population modeling for their treatment decision. And the patient population used in the model building is summarized in this slide. We modeled 50 patient data and tested several covariates to improve the predictability of the model. And here is the best model we built from the data. First, PK model was a linear three-compartment model for amubicin with one metabolite compartment for amubicinol. And we found out only amubicinol has a significant drug effect on neutrophil count, and it reduces the production of neutrophil cells with a long time delay. So four transit compartment was added to express the time delay. Furthermore, two different feedback mechanisms were added to the model, and both improved the fitness to data. Also, two covariates or patient background, body surface area, and patient performance status were selected in the covariate model for these parameters. Finally, the best PKPD model was successfully qualified through Bootstrap and Visual Predictive Check, and you can see the PPC plot in this slide here. And in the next step, we developed our Shiny app to execute Bayesian simulation based upon the best population model we selected. Since it's a bit tricky to run Phoenix NLME from our Shiny package, we used our RsNLME instead to implement the model because it has a great compatibility with our Shiny package. And I'm giving you several mock-ups of the Shiny app. Since it's still a kind of prototype, the user interfaces don't look so great, but it should be user-friendly and intuitive for physicians to input required information like dosing or observation and execute the simulation for their treatment decisions. Also, it can generate a data analysis report in Word format for their filing. And currently we are conducting a clinical trial to evaluate the clinical utility of this Shiny app right now. I mean, we are testing how well the population model could predict the transition of neutropenia and inform treatment decision, particularly for the necessity of GCSF administration. And I hope we can report the study result for you in the next 1 or 2 years in a kind of research paper. However, some of you might be wondering which software of Phoenix NLME and our RsNLME should be used for your own study. So let me address that concern in the next step. So we have Phoenix NLME and our RsNLME. Which one should be used? So first of all, both software is comparable in terms of the computational performance. So either of them could be used for any purposes, including a regulatory submission. However, in my view, if you are very eager to learn R coding for your career or familiar with it already, our RsNLME with Piranha could be a great tool for you. On the other hand, if you are a Win-Only user and need to comply with GXP requirements in your organization, Phoenix NLME might be a better choice for you as it works on the same platform to Win-Only and easily validated through a validation suite solution. So personally, our RsNLME is my favorite, and Certara has invested more resources to the development, but that's your choice. And please contact us if you need some advice on your decision. So our RsNLME could be a key tool for your data analysis on R. In addition to that, Certara has contributed to our community through the development of our packages and some educational courses for our programming. So these our packages were developed and used by our consultant in Certara for their own modeling and simulation project for a sponsor company. So the package quality has been proven by those experts already. For instance, GG Quick EDA provides a GUI to ggplot2 and Table1 packages. And CovEffectsPlot is another Shiny app to produce a forest plot to visualize covariate effects on a specific parameter. So there are the other useful packages available on this website as well, and you can install them for free. And our consulting team will continue to add another R package in the future, so it's highly recommended to bookmark the web page on your browser as soon as possible. So furthermore, if you look for some educational contents to learn our programming, we provide Certara R School content in the website as well, which provides a series of webinars for you to leverage our capabilities in your population modeling analysis. Okay. So I'd like to finish my presentation here. So I introduced our RsNLME solution today and some kind of R capabilities to help your drug development project. And with that, I'd be happy to get any questions from you. Thank you.
Thank you so much, Taka-san. I think a lot of questions were already.
Okay.
We might have a few more questions. We're just running short of time. But
yeah, sure.
There is just one question, I think, which is very important for us also as a distributor, which you can answer for all of us. Non-MEM is very popular, as you had mentioned. It's a gold standard. So is there any difference in the modeling result or between RsNLME and non-MEM? This is a very commonly asked question, and I think the audience will benefit from the answer.
Okay, yeah, thank you for the questions. Yeah, I agree with you. And the answer is actually yes and no. I mean, it's common to see some small differences when comparing outputs across different software like non-MEM and RsNLME. And the differences would change due to hardware and software and specific dataset and model characteristics. And this does not mean one tool is correct and the other is not. Rather, it's a nature of this type of modeling analysis. So I would recommend you need to check and decide whether the result can be trusted and make sense with you every time you model your data. And in this respect, RsNLME can provide you with a very useful diagnostic tool to evaluate your model. Okay, does that make sense?
Absolutely. Thank you.
Okay, thank you.
So much, Taka-san. Any other questions? We will email it to you. Thank you.
Course.
Yeah, thank you so much for taking the time today.
Yeah, okay, me too. Yeah, thanks so much to give me this valuable opportunity. I hope our software solution can help you your drug development project. Yeah, thank you, and have a good day.